Hey everybody, had a little bit of a problem with the previous link. I'm not really sure what happened there, but YouTube booted me off. So I am just sharing that link so that it can get all out to folks. Not really sure what happened there, but I'm here. I'm ready to hang and do some science, and you folks are welcome to jump in as we go. So, welcome to this week's episode of Kitchen Chemistry. As always, I am your host, Brianna, and we are back to do some at-home experiments. We're in the middle of our series right now where we're going back to the classics and doing some classic science venture experiments, ones that I grew up doing as a camper, and then now ones that I get to teach as an adult. So it's pretty fun. If you don't know what we're doing right now and you're like, hey, I don't remember this happening, check our previous episodes. They're all online for you to see. So, what are we doing today? Well, we're still talking about our homeboy yeast because yeast is such a good example, as I've talked about previously. It's the model organism for cell biology if we're talking about eukaryotes, and it's also a really good example of chemistry in eukaryotes. It's really easy to study, and I can buy jars of it at the grocery store to use for this forever. So, without further ado, let's get started. So we're talking about yeast today, and we already know about yeast. We talked about cell biology a little bit. We've talked about DNA. But we're going to talk about a new part of cell biology. We're going to talk about something today called enzymes. Now, what are enzymes? Enzymes is just a fancy word for a protein that has a specific task involving making or breaking chemical bonds. So that was a lot of words. So let's talk about proteins first. So we're used to hearing the word protein when we're thinking of things like meat or legumes or dairy products. It's even in a lot of, it's in plants too. But it is a molecule or a series of molecules that help with building blocks for life. So kind of like DNA is the instructions. DNA are the instructions on how to make proteins. Then those proteins are made, which can help make everything else. So, proteins are kind of like DNA in that they're made of small little pieces. Like we had DNA was made out of nucleotides. Proteins are made out of amino acids. And there are more amino acids than there are nucleotides, and people are actually inventing new amino acids because they're a bigger molecule, they're easy to manipulate, so they can make artificial ones that we can then use in specific experiments, which is really, really cool. It's very cool research, very fun. But there are some classic ones in our body that we use every day. And that's also why we need to eat protein. It's really important because our body can only make and process certain amino acids. So some of them we have to get from food. We cannot just make them on our own. So if we need them to make a certain part of ourselves, if we need them to make a certain piece of cellular machinery, or you need a certain amino acid in order to make your hair nice, or whatever. There's lots of different cellular processes. You need to get that from your food. So that's why no matter what, if you eat meat, if you don't eat meat, vegan, whatever, gotta have that protein because it is muy importante. So, proteins have jobs. Again, we said they started like amino acids, and they're a lot like DNA. They start out kind of linear, and then because they're such a varied molecule, they're actually able to bind together in lots of weird new shapes, which let it interact with other molecules, kind of like a tiny machine. So some are just structural, like I said, but these things we're talking about, enzymes, are these special proteins that are folded up in a way that they can interact with molecules to either put them together and make a new molecule or break molecules apart into smaller pieces. Kind of like how we digest food. That's a lot of enzymes all the way from your mouth with your spit, all the way through your digestive tract, are different kinds of enzymes. So, in general, if you're looking at an enzyme, it often ends in ase. So we have amylase in our spit, which breaks down amylose sugars. We have peptase in your, someone's going to correct me. Enzymes specifically for breaking down proteins in your stomach, and you have them all the way through your digestive tract, like lipase helps break down lipids, and there's lots more specific ones, but there's lots of general ones in our digestive tract. So, there are also ones in our body that are important for breaking down things that we call toxins. So I am saying toxins in air quotes because I know there's a lot of it's kind of a buzzword in the media of what our body does. So that's my brother giggling in the background with headphones in. I think we're done. Okay. It's kind of a buzzword in the media because people say like, oh, you need to go on a juice cleanse to get rid of those toxins. Or you need to eat this special expensive mushroom powder from the Himalayas. Maybe there is one, I don't know, to clear toxins out of your body. That's a bunch of baloney because your body is full of these little enzymes, these little machines that break down these toxins for you. Your body's got it handled. It can do it. Just eat healthy and exercise, drink lots of water, and you're pretty much good to go. So, for example, our bodies have special enzymes to break things down like peroxides. So, if you've ever heard the word peroxide before, it's probably because we've used this before. This is my nice hydrogen peroxide. Just cover up the brand. This is my everyday hydrogen peroxide. It is a solution that's only 3% hydrogen peroxide and the rest is water. So it's a pretty weak mixture, but it's often used in wound cleaning. Kind of some arguments of whether or not it's good as a wound cleaner, which I'll get into. But it's often used for it, and I use it because, any port in a storm, really. I'd rather have an infection and some damaged tissue, or no infection and some damaged tissue than an infection, but, well, the tissue's going to get damaged if it's infected anyway. I digress. So, peroxides. Peroxides, it's any chemical that's got two oxygens next to each other in the molecule. So there's lots of different peroxides, but the most common one is our homeboy hydrogen peroxide, which is just HOOH is the molecule. It's like a water with an extra oxygen. So instead of H2O, it's H2O2. So it's kind of like the joke. Two people walk into a bar. One orders H2O. The other one says, I'll have H2O2. And they drink it and dies. It's because you can't drink this. Because our bodies don't like it. Because peroxides count as a toxin. So peroxides can actually damage cells. We're not going to go into the nitty-gritty of how they break down cells, because we'd have to start talking about lipid membranes and lots of nitty-gritty cell biology. And I would need like a projector or a whiteboard, and there'd be a lot of arm waving and be here for a couple hours. So we're just going to say they hurt cells. So what our bodies do is so that they can't just sit there and damage cells, is our bodies have special enzymes to break down peroxides. So when you are pouring hydrogen peroxide on like a cut or something and it's fizzing, that is because there are exposed cells that are getting that peroxide into them, and your body's like, whoa, we don't like that, that's damaging our cells. So these enzymes go out and break apart the peroxides, turning it to water and oxygen. Just takes out that extra oxygen, and then we have H2O, which is our water, and then we just have O2, which is our oxygen. Works great, works like a charm, and then our body is safe. So that's why you get fizzing. So if I poured it on my skin right now, I don't have any exposed tissue, so I probably wouldn't have any fizzing. But if I had like a big cut on my arm or something, an infected toenail perhaps, we'd see lots of fizzing from those exposed cells. And lots of different organisms have this. This isn't just a human adaptation. I use humans a lot as an example because I'm one, you're probably one. Most of the people around you are a human, I'm assuming. So it's a good example because it's something topical. But lots of organisms have enzymes to combat these toxins. Lots of plants have them, which is really, really cool, to sometimes combat different toxins from insects, which is dope. And lots of animals have them too. It's how some animals are often like these enzymes it's how animals who eat something that maybe other organisms find poisonous, how they're able to eat it because they can often break down those toxic compounds before they become an issue, which is pretty darn neat. So to demonstrate how peroxides work, we're going to do two things today. One is I'm just going to do a speedy little demo to show you how there's different amounts of these peroxide destroying enzymes in different organisms. Not going to jump into myself. Don't you worry. And then we are going to do a good old classic experiment from Science Venture that I have been able to experience since probably 2003 when I first started on summer camp. That dates me a little bit, but that's fine. That I have enjoyed from 2003 straight through to 2020. So 17 years of experience with my good old hydrogen peroxide. So we're going to do this experiment together today, and the lesson plan will be up sometime within the next week, so you can do it at home, and it'll have all the information regarding peroxides, enzymes. If you're like, hey, I'm talking really fast, I know I do. That's how it goes. So let us go to the desk cam to see, or the table cam, to see what we are using today. So what are our materials? First of all, we always have a little baking tray because we want to protect our surfaces. I always use my oldest, grossest ones from Mother. Doesn't get mad at me. What else do we have? We've got, whoops, sorry, we don't need to stare at my compost bin. I've got, just like last week, the good old baby syringe. Works really well for measuring small stuff out. Got a cup of water. We have our hydrogen peroxide, just like I said. My cell phone, which I'm pushing out of the way. Some food coloring. Yeast. I basically shouldn't even put it away at this point because we use it all the time. Dish soap. I've got some measuring cups. I also have this little dropper that I found in my cupboard. You know, sometimes you just got to go on a bathroom deep dive and you find all kinds of cool stuff to use for science. I've got some yeast on a plate. I have some chopped apple on a plate. I also have some potato on a plate. It's just over there. And because I am working at home, I don't have my regular science venture equipment. As I've said for the last 6 episodes. 6 weeks even. Happy 6 weeks. So I had to go, hey, I need something like a test tube or a volumetric flask like I used for today. So I looked around my house for something that was tall and skinny that was still clear that I could see through so that you could see the experiments. Best thing I could find was this flask that is used for something that I'm not going to say on the internet because we are trying to be child friendly. But if you have any kind of tall, skinny glass like this, that you could perhaps toast with on New Year's, if you catch my drift, this works great. This is what I ran it, and it works pretty well. It's good to get, unless you have a bunch of test tubes at home. I don't, so I'm working with what I have. But whatever you have, give it a go. So let's get started. What I'm going to do is I'm going to show you how our hydrogen peroxide reacts when we add it to different substances. So first thing, I got the humble potato. Mash them, boil them, stick them in a stew. So I have my potato on a plate. I'm going to take my little dropper, hydrogen peroxide. Love that child's proof cap. It's also apparently 22-year-old proof. Oh my gosh. I'm so glad we live streamed this so you all get to enjoy me struggling to take off a lid. Hallelujah, we got it off. Didn't have to ask my mom for help. I need a cup because I can't get the hydrogen peroxide out. Once again, don't you love it that I present this live so you get to see me be a goof? Okay, so pour the hydrogen peroxide in a cup. So I have better access to it. We can always dump it back afterwards. Now, sucking it up with the dropper. Beautiful. Let's see what happens. So, I'll hold it up to the camera so that you can see better. Mom, I'm going to buy you some more hydrogen peroxide after this because we use a lot. So, you should be able to see our potatoes are fizzing. Why are they fizzing, perhaps? It is because they contain these peroxide breaking down enzymes that are turning that peroxide into oxygen and water as we speak, just like we mentioned. So we've got a good amount of fizzing coming from the potato because potato cells don't really want peroxides either. Often things that can hurt us also hurt plants. So that one's our first one. Pretty neat. I think it's pretty cool. So I'm going to let that fizz off to the side. Now we have our humble apple. Again, if you decide to try this at home, you're more than welcome. You can even try different things in your fridge because I think that'd be kind of cool to compare. The best way to do it is to break them into smaller pieces because the more surface area your pieces have, the more exposed cells they're going to come in contact to, the hydrogen peroxide that is, and the more fizzing you'll have, which is more fun. More fizzing is always more fun. So we're back to the table cam. This is now our apple. So let's get some more peroxide in this bad boy. And let's give it. Now the apple tends to react a little less strongly. But there is, oh yeah, I can start seeing some fizzing. Now we can see it's not that much. So we can guess that our apple probably has less of this peroxide breaking down enzyme. So it's fizzing a little bit, but not that much. I could just see a couple bubbles. I don't know how well you can see. So we can deduce that our apple probably has less of this peroxide enzyme. We'll take this and put it on the side. Oh, here, we can compare. This is our potato, still merrily fizzing away. This is our apple. So, an interesting difference. Last but not least, for our little demo, is shockingly enough, we're going to go back and show you some yeast. So, let us see how the yeast does. And we can see as soon as I started adding it, we have fizzing. Like it is instantaneous, it reacts. It smells very yeasty, it's very foamy. It's gonna continue to foam, there's lots of bubbles. I don't know if you can hear it. I can, it's fizzing a lot. So you can see, again, this is why yeast is a model organism for lots of eukaryotes, things like people even, is because they react very similarly to our cells. So just like how our cells would fizz up if exposed to peroxide, this is the same reason the yeast cells fills up. It's because otherwise it would be a toxin and the yeast wants to survive. So, remember I said earlier, some people swear by hydrogen peroxide for wound cleaning and some are like, no, that's not great. Is even though you have these peroxide breaking down enzymes, they still are going to damage some of your tissue. So I'm not going to give you any advice here. Don't take my advice ever on anything unless it's like how to make bread, because that's the thing I know how to do. But when I'm talking about like health and well-being, ask your doctor. Go with what you know. But from actual science, I can tell you that peroxide does kill bacteria. Yay! That's what we want. But it also does break down your cells. That's why we react so strongly. If it didn't break down our cells, we wouldn't see any fizzing. It would just kill the bacteria and we'd carry on. So I still use hydrogen peroxide, but again, a lot of people say to use it, a lot of people say don't. Take what you will, ask your doctor. Don't quote me on things. I know how to make science experiments and make bread and bake stuff. I'm not a healthcare professional. I only have a degree in microbiology, so. Moving on for experiment time. I guess there was an experiment, but we're gonna do like a flashbang fun experiment because, like, you've met me. I like flashbang fun experiments. So, let us assemble the materials that we need. We need the hydrogen peroxide, which we have. We need our water. Sorry, I'm only seeing my nose. We need our water, soap, yeast, measuring spoon, a cup, and our tall tubular container. So let's try this. First thing we are going to do with our tubular container is we're going to fill it maybe a third of the way full of hydrogen peroxide. So again, I am using 3% hydrogen peroxide. It says it on the label, 3%. You do not need anything stronger to do this experiment. Don't get 30%, it will burn you. This is safe to have on your skin, for the most part, we've talked about it. You don't need anything stronger. Please don't use anything stronger. You can hurt yourself. And I'm flinging hydrogen peroxide anywhere. So this is why I don't even use the 30%, because I'm a maniac. Flinging stuff everywhere. I am still struggling, there we go, with the childproof safety cap. I'm gonna take this hydrogen peroxide and put it out of the way so I stop playing with it. Gone. So, remember hydrogen peroxide in our cup. Next thing we're going to do is we are going to give it a liberal squirt of dish soap. Like, you don't need to add too much. This is less of a measuring activity and more of a ratio activity. But you want an amount of dish soap. Give it a good stir. I'm going to give that spoon a rinse because I need to use it still. You know, it would make more sense if I grabbed multiple spoons, but Since when am I that prepared? Moving on. Next, we are going to add, for funsies, this isn't necessary for the experiment, but you've seen me every week, and every week I kind of get increasingly more weird with the amount of colors I'm wearing. So I add food coloring because it's fun. You don't have to, but like, why wouldn't you? So we're going to add some food coloring to this bad boy. I think I'm going to make it purple because I like purple. So I've got this fun neon green pack, or neon pack of food coloring, which is sweet. Add lots because I want it pretty. It's going to be pretty dark in color, but don't worry, it lightens up. The more the merrier. So I added some pink, and I'm going to add a little bit of blue. You can see it's gotten nice and dark. If you're choosing to use color, you want more than you actually think you're going to need. You will see why in a few minutes. So, to recap, in our container, we have hydrogen peroxide, about 2/3, halfway full. A hefty amount of soap, probably about a tablespoon and a half, and some food coloring. Take this, I'm going to move it out of the way. Next step, we have some water. I actually think this is too much water. I probably only need like 3/4 of a cup of water, so I'm going to dump a little bit out. This is why we do it right by my sink, because it's a lot easier. So we have our cup of water. To our cup of water, we are going to add our favorite in kitchen chemistry, yeast. Good old baker's yeast. And we are going to add a teaspoon. So we're going to take our spoon, measure it out. Again, this is more about ratios than measurement, so don't be fussed if you get a little less or a little more than a teaspoon. We're going to add it to water. And we're going to want to give our water and yeast mixture a nice liberal stir. And I'm going to keep stirring that while I talk. So just like I talked about with bread, we need to, our yeast is currently kind of like it's in stasis. It's asleep, it's hibernating, whatever you want to say. The yeast is inert right now, it's not active. So in order to activate it, it needs water and often sugar if you want it to really thrive. But you can also activate yeast with just water. That's how, again, we made our sourdough, is we never added any sugar to it. We just activated with water. So we did add flour to it, which contains sugar, but I digress. Right now, you don't need sugar. That's the moral of the story. And I'm going to keep stirring my yeast mixture until, I like to say, it kind of looks like chocolate milk. By that, I just mean that it's like kind of opaque and cloudy and most of the yeast chunks are gone because A, I don't drink milk because I'm lactose intolerant, and B, this would be like the nastiest chocolate milk you've ever seen. More of what I look for when I'm stirring it is, again, no clumps. It's like milk cakes. No clumps. And it should smell yeasty. So I can give it, I'm not going to sniff it. We're having a brief interlude because my mother's phone is going off. She's vacated the premises. We may continue. Sorry. I live in a house with not a lot of walls, so we all kind of share the space. Often I have my family making funny faces at me on the screen over behind my laptop. So you ever see me laugh for no reason? That's probably why. So I will show you how well this is stirred. So it's stirred pretty well. There's still a little bit of like yeast floaties, but there's only so much we can do. Yeah, I'm going to scoop that out. It's kind of gross and slimy. It reminds me of pond scum. Oh, Pippin! Pippin! Do you want to say hi? Here. I'm gonna give you a treat if she actually listens to me. Oh, never mind. My dog was wondering what I was doing. She just walked away. So I'm gonna keep going. Sorry, gang. So last step, and this is where it gets messy, and this is why we use a tray. When in doubt, use a tray. I feel like I need that on a t-shirt. We are going to add our stirred yeast water mixture to this tube. Do not hold onto the tube unless you want to get messy. All of this is pretty safe once it reacts, but I recommend letting it do its thing before you play with it. So I'm going to count down from 3, and then I'm going to pour, okay? Okay. We're ready to go. 1, 2, 3, go. And it's alive! I know it spilled over, but I had to add lots. And we can see it's fizzing. So, and it's making some foam. So we have here is we have, kneeling on my floor so you can see me. What we have here is our yeast and our hydrogen peroxide are reacting, just like we saw earlier. So the yeast enzymes are breaking down the peroxide from its toxic form to its non-toxic parts. So it is breaking it down. It's breaking H2O2 down into H2O and O2. So that's water and oxygen. So normally that would just make gas and some water, and we don't really see the gas because it's gas. But what's happening is because we added our friend dish soap, is dish soap helps catch that air because it's more viscous. It's just kind of the nature of soap. Like when you rub it around your hands, you make some foam, bubble baths in the tub, same kind of idea. So when we add our soap mixture into our yeast hydrogen peroxide mixture, it traps all of that oxygen as it's made, which makes this sweet foam. And it's really fun to play with. So this is why I added lots of food coloring, because as you can see, it's like barely purple, because it's made a lot more foam than what was originally there. And because there's foam and there's lots of bubbles, it actually makes the light refract differently. Bit of a stutter. Light refract differently, so it looks lighter. But eventually, as the bubbles pop and the liquid condenses back into a regular liquid, because eventually the bubbles will pop, it's going to look darker. But in the meantime, we just have, as it reacts, we have water, oxygen, soap, and yeast, which means that it's perfectly safe to play with and poke. You can poke it with a spoon. It's pretty fun. I made this with my family, and we just made it on trays, and everyone was making like foam angels with their spoon. You can scoop it off the top. Kind of looks like a milkshake right now. Pretty fun. And we'll see, it'll just fizz right back up. And this reaction is going to keep going until one of two things happens. Either the yeast breaks down all the peroxide, or there's too much peroxide to yeast, and it'll either kill the yeast, or there's not enough enzymes to fully break down all the peroxide. So eventually, this experiment will come to a stop. I think it's almost there because it's going to stop. It's stopped overflowing. Like I can still scoop the foam out, but it's not overflowing anymore, so it's slowing down. But it gives us all a very fun foam to play with. This is a great activity to do with little ones because they can like fully put their hands in it. It's pretty safe. I recommend goggles to keep it from getting in your eyes, maybe like a smock or lab coat from getting it on your clothes. It's a really fun one to do. So I hope you enjoyed joining me today on this episode of Kitchen Chemistry, episode 6. I think we're about halfway through the summer now. Probably going to do 12 episodes total unless we're able to carry on to the fall. Fingers crossed we can. But here, I'll stand up. Thank you so much for joining me today on another episode of Kitchen Chemistry. Next week, we're going to do one more classic experiment. Then we're going to wrap up and go back to cooking. And I think we're going to let you in on a secret. I think our next cooking theme, because we did yeast, the next theme is going to be eggs. All about eggs. Believe me, we will eat them. And sugar and chocolate and butter and flour will be involved. Dun, dun, dun. So I hope to see you next week. Thank you so much for joining me. I will see you next Wednesday. Bye-bye.